All right, we have about 100 participants who have joined. So I think I will slowly get started here. So welcome everyone. It gives me great pleasure to introduce our speakers today. Today we'll have Dr. Miguel Ramalho-Santos, who has a long-standing interest in developmental epigenetics. He obtained his master's degree from the University of Coimbra, his Ph.D. from Harvard University in 2002, and subsequently became a UCSF fellow in 2003. Then assistant professor in 2007, associate professor still at UCSF in 2013. He joined the Lunenfeld-Tanenbaum Research Institute as a senior investigator in 2018 and is a professor in the Department of Molecular Genetics at the University of Toronto. Miguel has received several awards for his work, including a 2008 NIH New Innovator Award, the Royan International Research Award in Reproductive Genetics in 2016, and he was also the recipient of a Canada 150 Research Chair. He mentioned that I didn't have to say this, but I think it's pretty cool, so I'll just bring it up. While reading on Miguel's bio, I got to learn a few things about him that I didn't know. So he's not only an outstanding scientist, but he also has an artistic side. So in another life, he was part of a professional theater company. He was a drummer in a couple of rock bands, and he has published some creative writing work as well, which is pretty neat. Our second speaker after that is going to be Lamisa Mizan, who is a doctoral candidate in Miguel's lab. She obtained her bachelor's degree from the University of Toronto and is now working towards her degree within the Department of Molecular Genetics. Lamisa is particularly interested on the role of transposon interaction networks early in embryonic developments. So with that, we'll start with Miguel, who will tell us about epigenetic regulation of development in sickness and in health. And we have a slide up right now on an ATAC-seq workshop. If anyone wants a little bit more information on that, you can contact info@epigenome.ca. So with that, I'll leave it to you, Miguel.
Thank you. Thank you, Eric. Yeah, thanks a lot for the invitation. I'm really excited to present our work to you today, me and Lamisa. And we look forward to feedback. We're going to talk about quite a bit of unpublished work, and so we look forward to any suggestions you might have. My lab is interested in the epigenetic regulation of development, and today I will give you an overview, really, of what the lab is about, and leave sort of a more specific, more detailed project for Lamisa to describe after my presentation. So before we start, though, I would like to acknowledge that we, as the Santos Lab, acknowledge that the land on which we work is the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples. We know that this land and its resources were taken against the will of Indigenous peoples or without full respect for treaties. We have regular lab meetings on anti-racism and anti-bias, including on the history and present state of discrimination against Indigenous peoples in Canada, and what actions we as scientists and citizens can take to address that. We definitely welcome suggestions on these efforts and what we can do moving forward. So as I mentioned, we are interested in environment-genome interactions during development and inheritance. And I don't think it's too complicated to get someone fascinated like myself early on as a graduate student in developmental biology, in particular this process that goes from fertilization to the generation of the embryo and the fetus, and how you envelop this potential to give rise to all cell types of the body in such an organized fashion in space and time during development. Just as fascinating as that, I believe, is also how you can package the information to give rise to the whole magic all over again through the process of reproduction through the gametes. And as we know, the blueprint that guides a lot of development and reproduction is the genome. but more and more we appreciate that the environment provides important inputs, not just bad inputs, but also essential inputs for the unfolding of development and reproduction. And the areas that this research impacts are quite diverse, from infertility, of course, from studying fertilization and early development, to birth defects that can occur when errors are occurring during development. Also, as you know, there's a lot of research aimed at taking advantage of information gained from early development, pluripotent stem cells, and use that information to treat degenerative diseases, where particularly cell types of interest can be generated in the lab for further studies and even cell therapies. And also, we're learning that a lot of the processes that occur during development are hijacked during cancer when cells grow uncontrollably. I would add to this that maybe we're not always thinking about this particular angle, which is disease programming. The environmental inputs into development have now been appreciated more and more to be able to leave marks on the embryo that then when it grows up can have detrimental effects or impacts on its physiology and disease propensity, including potentially intergenerationally. So we're interested in all these different aspects. To come down to reality, then what we do is use a lot of mouse developmental biology and genetics, as well as mouse and human embryonic stem cells. So these are the models that we use. And Lamisa, after my presentation, a graduate student in the lab, is going to talk about our work on the role of transposons in early development. And I'm just going to leave that for her. I'm very fascinated by that work and her work and others in the lab, but she'll tell you about that. We have an ongoing interest for a long time now on the chromatin and transcriptional networks of pluripotency. I'll summarize some of that work. I'll tell you a little bit about our work on chromatin reprogramming in the germline, particularly as it pertains to intergenerational effects that I mentioned. And we have an interest on the regulation of stem and progenitor cells postnatally in adult stages and their connection to cancer development. So I'll give you just some snapshots. And of course, I want to remind you that more and more, we are interested in bringing in the environment as a variable. So we as biologists like to insulate things and not bring in the complication of the outside world to our developmental and other models. We're trying to flip that around and welcome that input. All right, I want to start from the very beginning, which is the principle of cell differentiation, to introduce a topic that is pervasive in the work that we are doing. As you know, all the cells pretty much have the same genome in our body, and it's really differential gene expression, by and large, that makes cell different. It's a gross simplification, but I think we can go with that. So if you take embryonic stem cells that differentiate into different cell types, and each of these cell types, including the embryonic stem cells, will have a specific transcriptional program that I'm simplifying here. So you have your housekeeping genes expressed pretty much in all cell types, and then there's tissue-specific programs in the stem cells and in each of differentiated cells that we can actually use to identify or diagnose what the cell is in many instances. What we've realized a few years ago is that stem cells can go into this state that we call hypertranscription, where they're highly activating their global transcriptomics. In other words, that they're really upregulating thousands of genes in their genome that they are expressing. So this includes the housekeeping genes as well as the embryonic stem cell program. It's not just a random willy-nilly upregulation of everything. They still maintain silence the programs that need to be silenced in stem cells. But what you also have to appreciate is that housekeeping genes are a vast majority of the transcriptome of any cell. And so this is a huge investment or effort that the cell is making when it goes into hypertranscription. And so I'll come back to where we study hypertranscription, but I want to highlight to you that this is often missed in even current standard analysis of looking at transcription in transcriptional profiling or RNA sequencing. So when a cell is in hypertranscription, this is the pattern that you have. Other cells have a lower level of transcription. But when we normalize the data, as we usually do for housekeeping genes, and of course, many of you can remember doing that maybe from your undergrad years to qRT-PCR for housekeeping genes, or even in Western blot, you can do that. We do that in RNA sequencing, many different techniques. What that does is it brings down the global transcription. It basically masks hypertranscription in the stem cells. So we need to come up with new ways, and that's what we've been doing in the past few years, to do what we call cell number normalized RNA sequencing analysis, where we add, for example, equal numbers of cells, and we spike in exogenous RNAs in the case of RNA-seq, but there's many other ways that this can be done so that we preserve these global transcriptional dynamics. And importantly, this is not mutually exclusive with the usual way of normalizing data to look at the outliers. We can still do that analysis just the same. And so we have been very interested in hypertranscription during development for a few years, like I mentioned. Maybe I'll try to find the pointer. Oops. The pointer.
We can actually see your cursor.
You can see the cursor? Okay,
yes.
So I'll just use that. So let me get rid of that. Okay, and oh dear, now I've done more than I wanted to do. Excuse me. I'll get rid of that. Okay, so zygotic genome activation happens during early development. Lamisa will actually talk more about that. So I'll skip that, but it's an important case, of course, because that's when the genome gets to be transcribed for the first time. It's a very special case of hypertranscription. We've been doing quite a bit of work in embryonic stem cells, as well as the peri-implantation embryo going from the blastocyst to post-implantation stages. We've documented hypertranscription also in primordial germ cells. These are sort of the mother cells of the germline that will in the future become sperm and eggs, and we'll come back to these cells in a minute. And we've also documented hypertranscription as being essential for the emergence of hematopoietic stem and progenitor cells at mid-gestation in the embryo. We have a lot of work ongoing on what are the molecular sort of nuts and bolts of the regulation of hypertranscription. So we have quite a bit of interest on a particular chromatin remodeler called CHT1 that facilitates the activity of POL2. There's a number of transcription factors that we and others have been interested in with regards to this notion of hypertranscription or transcriptional amplification. Important one that I'll come back to later is the MYC family of transcription factors. And I'm already hinting at here to you that a number of external clues can and do impact the levels of nascent transcription. And so this could be highly sensitive to environmental inputs if we start to sort of extrapolate into those layers. So more recently, though, we've been asking the question ourselves, how about adult cells? All this work that I'm describing to you is work that we've done over the years in different progenitor cells of the embryos. So how about in adult cells? And we thought that this would be a simple question to address early on, very naively, because there's so much now single-cell RNA sequencing data, and we generated data sets of our own. And we thought, well, that doesn't need to be cell number normalized. You just have single-cell transcripts, transcriptomics. And so that should preserve hypertranscription. Turns out that it doesn't, given the noise in single-cell RNA-seq data. The standard normalization method, which is global scaling, basically still assumes that each cell expresses about the same amount of RNA per cell. So to reduce noise, we're going to compress these global shifts in transcription per cell. That's what I mentioned is called global scaling. But in the lab, soon after the lab actually moved to Toronto, Yoon-Kyu Kim, an undergraduate student at the time, decided to flip this around and go investigate single-cell RNA sequencing data and see if we could use either spike-in data, like we've been using in bulk RNA sequencing data, to exogenously normalize the data, or using what are called UMIs, unique molecular identifiers, that count individual transcripts, and see, is it possible to look at what we call absolute scaling using RCCs or UMIs, also still get these different states that you get in these typical UMAPs of RNA sequencing, but now be able to capture still information of global transcript contents? Or is it just going to be a mess because, again, we're taking away some of that normalization? And it turns out that this actually works pretty well. And the immediate observation from Yoon's really fascinating work was that there are a number of settings in which he looked in adult organs. The stem progenitor cells are in a state of hypertranscription compared to more differentiated cells. So to illustrate the point, here's the bone marrow, and I'll summarize to you other organs that we've looked at. All the stem progenitor cells are in the highest content of transcripts per cell. These are these sort of pinkish cells over here. If you look at the same cluster at the levels of transcript, when we do this absolute scaling, you can see that they map very nicely. with the stem and progenitor cells, the one with the highest levels. And we can even look when there's enough depth in single-cell RNA sequencing at intron reads, which we're using here to infer nascent transcription. We have other ways in which we look directly at nascent transcription. And again, you can see that the stem and progenitor cells are elevated in their nascent transcription. So this was fascinating to us. We've looked at, like I said, in many other organs. And I want to give you a snapshot that might sort of get you thinking about what hypertranscription might mean in stem cells. Is this really just about the cells proliferating, for example, because it definitely is associated with proliferative compartments in adult organs. So this is a very interesting study that was carried out with muscle injury to trigger regeneration. So many of you might know that when you do injury of the muscle, what happens is that there's muscle stem cells called satellite stem cells that are usually quiescent, and when the muscle gets injured, they get activated to regenerate the skeletal muscle in this case. And immediately what we saw is that the muscle stem cells had indeed the highest transcript content of all the cells analyzed here in the muscle. But importantly, there was a very fascinating, to me at least, dynamics to the evolution of the transcripts. If you actually look just at the muscle stem cells, in the non-injured muscle, the stem cells are there, they're quiescent, and their transcriptional output is actually rather low. When you have this days post-injury, so half a day post-injury, there's a very rapid increase in the transcript content of the cells. It stays up and then it comes down as regeneration proceeds. But you can see that hypertranscription, or at least the global elevation of transcriptional content in the muscle stem cells, precedes the increase in muscle stem cell number by about a couple of days. So they actually, when they're the rather low numbers, that they're hypertranscribing the most. So it seems like it's anticipating the rapid increase in cell number that will be required for the cells to then fuel regeneration of the muscle. So this is a summary of this work. I encourage you to look at the paper that came out this year. Basically, I described to you the methodology that we use. We require this absolute scaling in single-cell RNA sequencing data. And like I said, I gave you the example of bone marrow and skeletal muscle. We've also looked at skin and intestine as other typical stem cell compartments in adulthood. And what we see every time we look at this is that the quiescent stem cells are actually in the low basal transcription. During activation, whether it's during normal renewal or regeneration, cells go into hypertranscription. And this is, yes, associated with rapid proliferation, but where we have time resolution, it's occurring prior to the rapid increase in cell number, and then it goes down in the differentiated cells, with some exceptions that we've found very interesting, and I can go into if you want to hear about them. Importantly, what we find is that a number, in fact, all of the pathways that we saw as diagnostic or recurrent in hypertranscription in developmental stages of hypertranscription, high levels of mTOR, MYC, chromatin remodelers, DNA repair, all these enriched pathways, including ribogenesis and translation, all the same pathways are recurring in adult stem cell, progenitor cell hypertranscription. So there seems to be a reutilization of this program in adult cells. And we suspect this is, of course, not just in normal physiology, but it is occurring, and others are reporting that it is occurring, including in disease settings, including cancer. So that was the snapshot that I wanted to give to you about how we use information from hypertranscription, sort of branch off to adult stages, and I encourage you to think about hypertranscription in your own work. I want to give you one snapshot of what we do with pluripotency, because there is a setting where we've studied hypertranscription, but this is actually the reverse of that. So a few years ago, what we found is that, yes, this is a stage where the embryos grow very rapidly, hypertranscription is occurring, and we're actually very actively investigating how it's occurring and what cells and what regulates them and to what extent it's actually happening. I won't have time to tell you about that. But we found a few years ago that this same pathway of the mTOR kinase, which is sort of a growth-promoting, nutrient-sensing pathway, if we inhibit it with catalytic inhibitors, we can very rapidly induce this paused state that actually mimics a natural state of embryonic pausing called diapause. In blastocysts in the lab, we can maintain them in the lab, pause in a reversible state, but we can also do that in embryonic stem cells, both in mouse and in human embryonic stem cells. And this basically suppresses everything that I've been telling you about, this hypertranscription, global translation, proliferation. The cells are just sort of sitting there in this paused pluripotent state, sort of a suspended animation state. They're not in G1 arrest. They're actually still cycling, but very, very, very slowly. And like I said, it's reversible. We can put the cells back into surrogate females, and they will give rise to as far as we can tell, normal fertile mice. So we reported that a few years ago, and these are the hallmarks. It's induced by environmental scarcity. So there you go. We start to see these connections to the environment that I've been hinting at. It involves reduced proliferation, lower levels of mTOR and MYC pathways, hypotranscription, hypotranslation. Metabolism is rewired in interesting ways towards autophagy. And like I said, it's a reversible state. So a few years ago, Evelyn Collignon, ex-postdoc that's now at the University of Brussels, joined the lab to ask specifically the question, how about post-transcriptional regulation, right? Because maybe it's a question that many of you have in your mind. Here I am sort of going on about hypertranscription and hypotranscription, and clearly that's just one piece of the puzzle. Something must happen post these levels of transcription to add additional layers of regulation. So Evelyn suspected that, and she had a keen eye to think about RNA methylation. She didn't go straight to look at M6A RNA methylation, which some of you might know is probably the best study on RNA methylation. She actually did an unbiased mass spec analysis of RNA modifications and found that M6A was increased in the positive state. So I'll give you a summary of what this is for those of you who don't know. It's very similar to the paradigm that we explore in DNA methylation. In other words, there's enzymes that go ahead and methylate particular residues in RNA, in this case, adenosine at the M6 position. There's also demethylases that can remove the methyl group. And there are readers, just like in DNA methylation, that can bind to the methylated group and impact a number of processes, of RNA processes, right? So having that information, what Evelyn sought out to understand is, what does M6A methylation do in paused pluripotency, both in mouse embryonic stem cells as well as in the blastocysts, using METL3 knockouts for her studies. I'm just, again, going to summarize this story because I don't have time and I'm going to give you a broad overview, but it's also recently published. So this is what we knew already. So if you inhibit mTOR with mTOR inhibitors, we induce this hypotranscription state and this diapause-like dormant state in blastocysts and embryonic stem cells. So what Evelyn found is that there's, in this paused state specifically, an increased chromatin recruitment of this METL3 enzyme that methylates M6A in RNA. And we have some clues as to why that is, but I'm not going to tell you about those today. What this does is that in the paused state, then there's an elevation, even though there's a global downregulation of many things, there's an elevation of M6A methylation in RNA. And what this triggers through specific M6A readers is an increase in RNA decay and downregulation. So not only do you have hypotranscription, you're also degrading more of the transcripts that you're making in the paused state. So that could have been it, but Evelyn decided to dig further and ask if particular mRNAs really need to be downregulated by this pathway for the cells to stay in this dormant state. And she found that that's, yes, the case. She found that it's really important to destabilize via the same process the mRNA of MYC-N or N-MYC, which is a major promoter of hypertranscription and proliferation. So if you downregulate N-MYC, what you get is hypotranscription, reduced proliferation. You maintain this paused state. And she proved this in a number of ways. I'll summarize the two key ways in which she did it. So if you knock out METL3, you disrupt pausing because now MYC is more stable. But if she knocked down MYC in those METL3 knockout ES cells, she restores pausing. So MYC really needs to be destabilized for the cells to stay in the pause state. And she went on to show that if you force demethylation just of the MYC-N mRNA in wild-type cells with a Cas-Rx demethylase fusion protein, if you stabilize the MYC-N mRNA in wild-type cells, this is sufficient to disrupt pausing. Again, saying that N-MYC is critical to be downregulated by this mechanism for the cells to stay in this dormant state. So as I mentioned, this came out recently. I encourage you to go and look at it. And I will tell you one more vignette, because now this is unpublished work before we move on to Lamisa's, to give a sense of what we like to do, what fascinates us about the germline and how it might be responsive to environmental factors. For that, I'll introduce very briefly, because I think everybody here knows what DNA methylation is. You know that it's the best studied, best known epigenetic mark, the addition of the methyl group, particularly in cytosine in mammals. It doesn't change the genetic code. You can still have all this nice pairing here. It's a very old methylation or epigenetic mark. It changes the activity of genes in ways that been studied for decades. And there was this quest of how this is removed. There's a few different ways in which DNA methylation can be removed, but importantly, it can be actively removed by TET enzymes that can initiate the process by hydroxylating and then further oxidizing the reaction product. So a few years ago, 10 years ago now, we showed that vitamin C is a key cofactor for TET enzymes to promote DNA demethylation, and that was in embryonic stem cells. And then more recently, what we showed is that this actually is important in vivo in mice. So what we showed is that normally in mice, vitamin C is important to be taken up basically by the fetus, so that in the germline, so in the primordial germ cells of mice, TET1 enzyme has its sort of full activity and demethylates, leading to the activation of germline genes that regulate germline development, also demethylates transposable elements, potentially as a defense mechanism against them. But what we found is that when you take away vitamin C from the diet of this particular mouse model of vitamin C deficiency, what we get is lower activity of TET enzymes and incomplete DNA demethylation. And this actually very nicely mimics the TET1 mutation. So this is removing vitamin C, mimicking the genetic mutation of TET1. There's an incomplete removal of DNA methylation. Like I said, this leads to defects in meiosis, development of the oocytes. And when this fetus grows up, which is now a normal mouse, we give back vitamin C at mid-gestation. It grows up to be totally fine as far as we can tell, but it's infertile or subfertile. So that leaves sort of a long-term impact of this deficit of vitamin C during gestation. So what we're asking in the lab right now is being a bit ambitious, but also having a bit of fun with models that we can implement is what else could the fetus sense and that can have long-term impact. So I've shown that for vitamin C. Well, how about things that really mammals have experienced over evolution and experience right now, including humans in many situations during pregnancy? How about starvation? How about fear, stress? About infection or exposure to different pathogens, cold, heat. I mean, we welcome suggestions. We can't do all of these, but it's been really fun to think about what might be potential environmental inputs that could have actually had an impact during evolution of mammals in these epigenetic readouts that may have had transgenerational consequences. So Giacomo started with starvation. Since we were going from one nutrient, he's now basically doing cyclic starvation in mice. And I don't have time to describe the whole data by any means. One day he will, I hope, in this forum. But what he's shown is that if you starve a pregnant female mice during some specific windows, I'll show in the next slide, that this has effects on the metabolism of F1 and on the metabolism F2. So these are sort of the grandchildren of the starved pregnant female. And these, of course, have never been exposed to starvation. And these defects in F2 are transmitted if we use the F1 sperm to basically to IVF. So in other words, the sperm carries the information that somehow is telling the F2 to have metabolic defects because their grandmother was starved during pregnancy. So this is the actual paradigm. We're not totally removing food from the mice the entire gestation by any means. It's basically 2 cycles of starvation during mid-gestation. Then we give back food at 13.5. And at this point, we take to birth and do those analyses that I didn't have time to describe to you, but I'll be glad to show any data if you want. Or we take primordial germ cells, because of course, that's these cells in green over here. These are already the precursors of the sperm and the eggs that are forming in these embryos. And we ask the question, if just like in the vitamin C, where we could see at the same stage, 13.5, the deficiency in vitamin C having an impact on the chromatin state, in that case, DNA methylation, and on the transcriptional state of primordial germ cells, how about in this case with this pattern of cyclic starvation? And what Giacomo found is very interesting and confusing, and we're still trying to understand it. So this is the RNA-seq of those green cells at the end of this cyclic starvation period. And what you can see here is basically there's a massive upregulation of genes in those PGCs. Even though there's less nutrients, they're actually in a state of hypertranscription. That's what this MA plot shows. There's upregulation of a number of growth and transcriptional pathways, deregulation of some metabolic genes that we're looking into, and it's a male-specific effect, so that this is just the male PGCs that are showing this hypertranscription. And well, we asked what more is going on with these PGCs. So at this point, we have a number of sort of ideas, but Giacomo started by doing a TAC-seq. to look at accessible chromatin, again, at these PGCs that have just been starved. And what we see is actually an increase in chromatin accessibility in these male PGCs. And if you look at the enrichment for particular pathways on the peaks that are more open in the PGCs from the starved cohort, you see actually a good validation that the cells are in hypertranscription, so positive regulation of transcription and regulation of transcription in general is highly enriched in these more accessible chromatin sites. And then we can go look for motifs. These are actually a number of interesting transcription factors that are present and active and important for primordial germ cells to develop, but also have roles in adulthood. So we're trying to understand, is there anything here that could potentially leave some sort of imprint that when these cells go on to become sperm, because it is passed on in the male germline, could they still have an impact in that F2 generation? So that's the question that's open for Giacomo to resolve. So these are the take-home messages from my talk. We continue to be very interested, and because we bump into hypertranscription left and right now, it's pervasive in stem and progenitor cells. It precedes proliferation during regeneration, and I encourage you to check it out in your data. I hope I convinced you that pausing is pretty cool, even though I didn't talk too much about it. The hypotranscription and RNA turnover both collaborate to ensure transcriptional dormancy of embryonic stem cells and blastocysts. And I didn't have time to tell you, but there's also a very interesting cancer angle here where these pathways are co-opted during cancer dormancy. Finally, I told you that mammalian embryo, I hope I convinced you that it's attuned to the outside world. So even though those germ cells are buried inside those developing gonads, inside the fetus, surrounded by a placenta and a uterus and a whole mom, they're not insulated from the external environment like sometimes we tend to think about mammals. They're actually, we think, actively sensing out the external environment, and that starved PGCs are in hypertranscription. It remains to be seen whether this is what triggers the phenotypes that we see in subsequent generations. So with that, I'll stop. Thank you very much. I mentioned the people that did the work. Primarily, I talked about Yoon Kyo Kim's, Evelyn Collignon's, and Giacomo Furlan's work in the lab. Thank our collaborators, and I'll just do one last plug for the LTRI. We've been actively recruiting for the past few years, and we have a new search also that just opened. This particular one focuses on muscle and bone, but in very, very broad sense. So it's a musculoskeletal chair. So I'll put it up there. Encourage anyone interested to apply, and I'll be glad to take any questions.
All right, thank you, Miguel. That was really interesting. So for the Q&A, we ask that you open the Q&A box in the Zoom menu and that you type in your questions in there. We'll then read them and allow the speakers to respond. And we already have a few coming in.
That's good. So I'll just read. There's an anonymous attendee that asks, what is the y-axis label? If you can be more specific about what slide or what was the experiment, I'm sorry if I mislabeled something. I could have, because I've simplified it a lot for this talk, but I'm not sure which one you're talking about. So maybe if you add some comment on that. Another question, thanks for summarizing the work. How pervasive do you think is hypertranscription in chronic disease? So I'll just answer that question. So we're starting to get, so the nice thing about single cell RNA sequencing data that finally convinced me that we should be doing some of it, and we're doing quite a lot of it now, is that it really starts to get us an opportunity to look more at dynamics at different stages, different timings during whatever organ we're looking at or whatever stages of development. So hypertranscription is quirky. It's not on all the time. Including in the hematopoietic stem and progenitor cells, including in the early embryo. So we think that there's peaks that, at least as far as we've looked at, anticipate stages of rapid growth, but then the cells can sort of take off on their own. So I think it's pervasive in cases where you have opportunities for either rapid growth or where the cells, for example, are secreting a lot of some factor, producing a lot of some protein. Those are situations in which we've also seen hypertranscription. So We have questions on, for example, in the setting of immunity and immune activation. Others have looked at B and T cells, for example, that enter a state of MYC-driven hypertranscription. So I'm sure there's going to be other opportunities to look at this, but we might need some sort of time resolution for those, just like in the muscle regeneration. Is it possible that hypertranscription alters other genes besides housekeeping genes in specific cell types? I think it's not only possible, I think it's likely. So the way we see hypertranscription, and it arises from the data, it's not just this linear, let's say every mRNA is now increased by twofold, and that's it. And then the genes that are silent are still silent. We see genes that go up by tenfold. We see genes that go up by only 1 point something fold. We even see genes that are going down in a state of hypertranscription. So there's some sort of logic to the madness of amplification. It is a global amplification of thousands of genes, but I think some genes might matter more than others, and that's sort of work ahead for us to figure out. It might be different in different cell types. And another question is, have you looked whether hypertranscription has an impact on the global 3D architecture of the genome? No, I'd love to. We haven't ventured into that yet for now, because we are trying to gain these higher resolutions, including with single cell data. We sort of decided to focus on that, but we've been able to extrapolate to some models, particularly in mouse embryonic stem cells, where we can now get, larger number of cells that might allow us to do experiments that we could otherwise do. And in that setting, we're thinking that we can start to add a number of other analyses of chromatin states, polymerase activity, including 3D architecture. We haven't gotten there yet. Oh, in muscle stem cells, it sounds like that was the question about the y-axis label. So I maybe can just quickly go back. Let's see. Oops. Well, that's going to take a while. So maybe it was here. Sorry, these were UMAP. There's probably UMAP 1 that I cropped over here. Is that what you're talking about? These are transcript contents. So these are counted with UMIs, and these are number of cells. So if you're asking about y-axis, that's what that would be. I'm not sure that answers the question, but looks like there's more. Maybe we should let Lamisa keep going.
Yeah, exactly. So we'll cut it here for now, but in the meantime,
Sounds good.
You'll be able to answer them by typing the answers.
Okay, I'll do that, and Lamisa can start her talk.
All right. So now we have Lamisa, who is going to tell us about line 1 and how it promotes nuclear compartmentalization to repress the 8-cell state in embryonic stem cells.
All right, thank you so much for the introduction. Like Dr. Campos mentioned, my name is Lamisa. I'm a graduate student in the Santos lab here at University of Toronto. And today I'll be presenting a lot of the work from the lab that's really been spearheaded and done by a postdoc in our lab, Dr. Juan Zhang. And together we've uncovered this role of line 1 in promoting nuclear compartmentalization to repress a key stage during embryonic development. And so to begin, we want to first introduce what line 1 is. So line 1 is a retrotransposon, which means that it can copy and paste itself all across the genome. And in fact, in human genomes, they encompass almost 20% of the total genomic content. However, even though historically retrotransposons in line 1 in particular has been regarded as this selfish or parasitic piece of DNA, our group, as well as others, have begun to uncover the actual functions that line 1 RNA could have in a variety of different contexts. And the context in which our lab is interested in uncovering these roles of line 1 is actually during development. And so previous work from our lab has shown that during mouse embryonic development, there's during this developmental trajectory, during the transition between totipotency and pluripotency, which occurs during the 2-cell stage, which is a very particular stage during development, and quite critical because it's also at the time when zygotic genome activation occurs. We found that mouse line 1 is actually required to repress this stage and is actually important for maintaining the stem cell and pluripotency in the mouse embryonic stem cells. And this work was primarily done by Dr. Michelle Perchard, a former postdoc in the lab. And she showed that when you knock down mouse line 1, we actually have an upregulation of the gene expression program that defines the two-cell state. And so we began to wonder whether this function of line 1 could be conserved in human embryonic development. So during the human embryo development, this zygotic genome activation, as well as transition from totipotency to pluripotency, occurs during the 8-cell embryo. And so we wondered whether line 1 could actually regulate this transition between totipotency to pluripotency during the 8-cell state of human embryonic development. And to ask this, we designed a antisense oligo-mediated knockdown system where we can specifically target the line 1 transcript in the human embryonic stem cells. And upon 48 hours of transfection of these antisense oligos, we're showing here that by RNA FISH, we actually have a depletion of line 1 RNA in the knockdown antisense oligos that target line 1 compared to a antisense control or the sense control. And so we performed RNA-seq in this line 1 knockdown condition in naive human embryonic stem cells. And first, we saw that overall, the transcriptome of the line 1 knockdown condition seems to be quite distinct from our control conditions. And then going into more of the gene expression profile, we saw interestingly that in our knockdown condition, there's actually a significant upregulation of key 8-cell stage markers compared to the control cells. And importantly, in order to validate that in an alternative or orthogonal knockdown approach of line 1, we designed a CRISPR-I approach in which a dCas9 that's fused with a CRAB transcriptional repressor, when targeted to line 1 at its 5' UTR or promoter region, there is also a significant induction of the 8C gene. So we can see by 2 different knockdown approaches that line 1 knockdown in these human embryonic stem cells can activate key 8-cell stage markers. So we were interested in figuring out how the fate of these cells change overall. And to do this, we performed also a single-cell RNA sequencing. And we could see that in our knockdown cells, there's actually a promotion of the generation of 8-cell-like cells in the human ESL culture. And this was further validated by two independent groups and gene lists of this 8-cell stage that are shown here on the right. And importantly, we could see that whereas in the control cells, there's only a very, very low 0.07% of 8-cell-like cells that are in culture. In our line 1 knockdown condition, we could see that it increases robustly to almost 5%, generating 8C-like cells. And so these results really suggested to us that line 1 RNA is acting as a barrier to the 8-cell program during human embryonic development. However, we don't know how this repression of the 8C program could be mediated by line 1. And so inspired by work that came out of different mouse studies, we looked into the idea that nuclear compartmentalization could be important for mediating these changes. And so to investigate this further, we performed 2 different sequencing approaches, lamina-associated domain sequencing, which is LAD-seq, as well as nucleolus-associated domain sequencing, which is NAD-seq. And using these 2 distinct kind of nuclear compartments, we wanted to analyze where the expression of line 1 could be coming from in relation to these nuclear compartments. And so analyzing both the LAD-seq and the NAD-seq, we saw that the line 1 family, as well as specifically young human-specific or primate-specific line 1s, are actually enriched in both the LADs and the NADs compartments compared to other repetitive elements. And we further validated this using a DNA FISH approach where line 1 DNA, labeled here in green, is shown to have a preferential association with both the nuclear compartment, with nucleolar compartment, which is labeled here in white by the nucleolin protein, as well as with the lamina compartment, which is labeled here in red with the lamin B1 protein. We also wanted to further see that the line 1 RNA itself, which is what we are hypothesizing the functional molecule, is preferentially localized to both of these compartments. And we validated this using an RNA FISH approach where we labeled line 1 RNA here in the white signal, and we could see that it is preferentially localized to the periphery of the nucleolus, as we were able to label the 45S RNA to show where the center of the nucleolar compartment is, as well as to the lamina, as we labeled here again with the lamin B1 receptor. We further validated this using a cross-linked RNA immunoprecipitation approach, where we performed pull downs of either nucleolin protein or lamin B1 protein, and we're able to show that the pull downs of both of these proteins show enriched association with line 1 RNA when compared to our negative control U1 snRNA. And so taking these together, we believe that line 1 does have a preferential association with these distinct nuclear compartments of the lamina and nucleolar domains. However, we still have to wonder, could this be the mechanism by which it can mediate the 8C gene association? And in order to do that, we went back to our RNA-seq data of line 1 knockdown and tried to figure out where the genes that are upregulated, specifically these 8C genes, could be localized. And we were surprised by this finding, where we looked at the on which chromosome majority of these genes that are induced by line 1 reside. And we're surprised to see that chromosome 19, compared to all the other chromosomes, were actually enriched with genes that were induced by the line 1 knockdown. And in particular, we noticed that several of the key 8C genes that demarcate this stage during embryonic development were also located on chromosome 19. What's even furthermore, what we were surprised by is when we did an analysis of nucleus-associated domains and lamina-associated domains across all of the chromosomes in the human cells, we were surprised to see that chromosome 19 actually has quite a unique profile of these nuclear compartments, where there's a enrichment of NADs on chromosome 19, similar to other chromosomes. However, there's a specific depletion of lamina-associated domains on chromosome 19. And so this unique profile of chromosome 19, in conjunction with the idea that many of the genes are upregulated, including the 8C genes, are located on chromosome 19, we hypothesized that there could be a relationship between nucleolus-associated domains and chromosome 19 that could be mediating the gene expression changes that we see in the 8-cell stage. And so in order to test this, we took a DNA fish approach, where we used chromosome 19, chromosome painting, to label chromosome 19 DNA and see its association with the nucleolus, here labeled by nucleolin protein. And so in naive human ES cells, we see that there is indeed an association between chromosome 19 as well as the nucleolus. However, we were surprised to find that in 8C-like cells, there's actually a decrease in this association between the whole chromosome and the nucleolus. And this is quantified here on the graph on the right. And so what these results kind of suggested to us is that the association of nucleolar domains could actually be the regulation, gene expression regulation mechanism that we see to correspond with the changes in gene expression in these 8C-like cells. And so we were interested in finding out more about how the nucleolar domains could be organized and how line 1 could be mediating these changes in nucleolus. And so we performed two separate ways of perturbing the nucleolus in order to see how this might impact the generation of 8C-like cells. The first way is through the canonical RNA pull-1 inhibition. And so we performed 8 hours of BMH treatment compared to DMSO. And we saw in these naive human ES cells that the morphology of the nucleolus changed to a rounder nucleolus shape. And by RT-qPCR, we were able to see that just perturbing this perturbing the nucleolus through pull-1 inhibition was also able to strongly induce the upregulation of these 8C genes. As an orthogonal approach, we also adapted our CRISPR-I system in order to knock down a core protein of the nucleolus, nucleolin, and we were able to see similarly an upregulation of these 8C genes. And so taken together, we do believe that these nucleolar domains can repress these 8C gene expression that we see in human embryonic stem cells. So taking this back to our line 1 studies, we found that in our knockdown line 1 cells, we analyzed how the nuclear architecture might be changed, and found that line 1 RNA could actually be required for the maintenance of the nuclear architecture. When looking at the control cells, we see a wild type nuclear morphology where the outer ring, which is marked in green here by nucleolin protein, encompasses the fibril center of the nucleolus, which is marked here in red by fibrillary protein. However, in our line 1 knockdown, this fibril center is actually expanding, and this leads to a stressed nucleolar morphology. And so these results suggest to us that line 1 RNA is actually required for the maintenance of nuclear architecture. And so Inspired by these findings, ongoing projects in our lab have started to now begin to probe more how nuclear architecture might be mediated. And one of the core findings that we came across in the literature is that these nucleolus organizer regions, or NORs, which is essentially where the rDNA arrays in the human genome are arranged across the 5 acrocentric chromosomes, could actually be mediating this architecture. And so we became interested, inspired by the work of the Mixed lab, in these sequences called the distal junctions. And these are sequences that are present across all 5 of the chromosomes, and actually could be functional in organizing the nucleolus. And so we performed DNA fish of these distal junctions in the naive human ES cells, and we saw that these DJs actually localize to the nucleolar periphery, similar with other cell types that have been tested in the literature. And so this suggests that its localization to the perinucleolus suggests that maybe it could be required or have some function in the organization of these NORs. And so we dug further into these distal junction sequences, and we're surprised to find that embedded within these DJs across all 5 of the NORs are actually a full-length line 1 element that corresponds to a young active line 1 family. And what's even further is that we were able to validate in 2 separate through 2 separate assays that these line 1 elements are also highly expressed in the naive human ES cells. Using K4 trimethylation chip, we were able to show that in the naive K4 trimethylation chip compared to the prime cells, there's actually a stronger enrichment at these distal junction embedded line 1 specifically. As well, we use RT-qPCR to validate that the expression of this distal junction line 1 is highly expressed in 3 different media conditions of these naive cells compared to the prime cells or differentiated cell types. And so some of the ongoing work that we have in the lab is to investigate further the putative cis regulatory functions of this distal junction embedded line 1 at the nucleolus. So we were first able to confirm that this line 1 RNA is actually retained in cis at the nucleolus using, again, this cross-linked RNA immunoprecipitation with nucleolin pull down, where we could see a strong enrichment of these distal junction embedded line 1s compared to our negative control RNA. As well, we adapted our CRISPR-I system in order to specifically repress transcription of our distal junction line 1s, and we're able to confirm that we have efficient knockdown of the distal junction line 1s and not targeting any global line 1s elsewhere in the genome, as validated by the 3rd primer here. And finally, some of the future experiments that we have going on is through preliminary qPCRs, we were able to see that just knockdown of these distal junction line 1s that are at the nucleolus, we also have upregulation of the 8C program in the naive cells. And so we'll be performing a more transcriptome-wide analysis with RNA-seq, as well as doing future nucleolar structure and functional analysis, as well as analyzing how the chromatin changes might be mediated. Finally, our collaborators in Mixed lab will be studying the function of this line 1 further through CRISPR deletions in a specific cell type. And so to summarize the work that Juan and myself, and as well as other members of the lab, have been working towards, our model is that line 1 is actually required for nuclear compartmentalization in order to repress the 8C program. So in naive human ES cells, we see that there's an irregular nucleolus kind of morphology, whereas in 8C-like cells, there's more of a round-shaped nucleolus. And as this transition from totipotency to pluripotency occurs, the chromosome 19, which houses many of the 8C genes, is actually increasing contacts with this nucleolar domain. And importantly, when we knock down line 1 and we are able to see an induction of these 8C-like cells, we see a disruption of the nucleolar structure, which could be the potential mechanism for which there's an increase in gene expression of those that are located on chromosome 19 and associated with the nucleolus. And some of the work that I wasn't able to talk about today is more detail and resolution into putative activators of the 8C stage, which are genes such as TPRX1 and H3XY, which have been, other groups have also postulated as activators of this core 8-cell stage. We were able to do more resolution studies on how these 2 different TPRX1 and HDXY have preferential interactions and loss of interactions with these nuclear compartments. And so to summarize, we believe that as the human embryo is going through these different stages and transitioning between totipotency and pluripotency, there's a maturation of subnuclear compartments and a corresponding establishment of nuclear compartments such as LADs and NADs. And line 1 is a core player in regulating the compartmentalization of this gene expression program. And with that, I'd like to thank the whole lab for all of their support. Particularly, I want to emphasize a lot of this work was really spearheaded, and Juan Zhang, our postdoc in the lab, is a big part of this project, as well as Dr. Kirti Mittal, Kevin, and Shaquille, who helped with a lot of the imaging. And finally, I'd love to thank our collaborators and everybody around, really around the world that have been involved with this project.
All right, thank you, Lamisa. That was also very nice. And I see that we already have a few questions here, starting with Michael Lee. Do you want me to read it, or do you want to read it?
Yeah, I kind of lost the floating bar, but let me try to get it back. Okay, I see it now. Okay, so the question asks, do you think line 1 RNA is acting in cis or in trans in its compartmentalization function? So there's a lot of line 1, there's a lot of different genomic locations of line 1. And so it's hard to know for sure if it's the line 1 RNA that we're studying at the nucleolus was produced from the genomic source is at the nucleus as well. However, there is some few evidences that it is a cis function. For example, in mouse data, when you perform a chirp of line 1 RNA, majority of it does pull down its own DNA. So it does suggest that there is more of a cis regulatory function. But I think to test that for certain, one of the things that could help is if we were able to, specifically label, for example, with the distal junction line 1, if you're able to specifically label a line 1 where we know it's transcribed from and follow it in the cell through imaging, that could be one way to know for sure if it's cis or acting in trans. The next question asked, maybe I missed this, is nuclear architecture normally different pre versus post 8C stage? Yes, so the nucleolus, the maturation of the nucleolus is something that's dynamic throughout development. So in other works, even in mouse data, there is changes that occurs in the nuclear architecture as it's progressing through different stages of pluripotency. Next, how does line 1 knockdown affect the tri-layer structure integrity of the nucleolus and also heterochromatin surrounding the nucleolus? So in our line 1 knockdown cells, we saw that there was actually a disruption of that, the 3 components of the nucleolus, and we saw that there was actually an expansion of the center, the fibril center, which is normally where the RNA pol 1 transcription occurs. So more of the processing and ribosome assembly occur in the outer rings in the granular layers. And so we see a disruption of that tri-layer structure, and that could correlate to the disruption of the nucleolar, the heterochromatin that surrounds the perinucleus as well.
And I believe we have one last question here.
And the question is, do you intend to study other transposable elements in a similar manner? Definitely, I think that line 1 is not there's many transposable elements that are highly expressed specifically in embryonic stem cells or throughout development. So if we have I think there is definitely more to be uncovered, especially with HERV RNAs as well as other like repetitive satellite RNAs as well. So potentially more work coming out of that, coming from there soon.
Yeah, I'd say plenty of more angles and work to tackle there. All right, so we're smack on time. So we'll end here. And again, I'd like to thank our 2 speakers. This was super interesting. I learned quite a bit here. And yeah, round of applause.
Thank you, everybody.
Thank you very much.
Thank you for all the great questions.